But today I'm looking forward to sharing with you some approaches to measuring blood-brain barrier breakdown in Alzheimer's disease and to share some of our preliminary data on this topic. Okay, so I expect that most of you in the audience are quite familiar with Alzheimer's disease. But for those of you who may not be familiar, I'd like to provide just a little bit of context. So Alzheimer's disease is an age-related neurodegenerative disease that ultimately leads to memory impairment and dementia. It's characterized by several hallmark pathological changes, which start with the accumulation of extracellular amyloid beta plaques. And this is thought to be the first pathological change in Alzheimer's disease occurring several decades before the appearance of symptoms. This is followed by the accumulation of intracellular tau neurofibrillary tangles. And together, these co-pathologies, the amyloid and the tau, are thought to lead to neurodegeneration, which can be detected on a brain MRI as brain atrophy or brain shrinkage. This neurodegeneration then leads to decline in memory, followed by decline in other cognitive domains, and then ultimately impaired activities of daily living, which is the definition of dementia. Now, recently, it's become increasingly recognized that the vascular system likely plays an important role in the pathogenesis of Alzheimer's disease. And I hope you can appreciate from this beautiful illustration here just how important the vascular system is to the brain. The brain is very highly vascularized, and this extensive network of blood vessels is critical to provide the brain with oxygen and nutrients and to fuel neural activity. Now, why is this relevant to Alzheimer's disease? Well, up to 80% of individuals with pathologically confirmed Alzheimer's disease have been found to have vascular co-pathology. In addition, several vascular health risk factors, such as hypertension, diabetes, and obesity, are among the strongest modifiable risk factors for Alzheimer's disease. So there appears to be a very close relationship between vascular risk, vascular dysfunction, vascular pathology, and Alzheimer's disease. Together, this raises the question, where exactly does vascular dysfunction fit into our model of Alzheimer's disease progression and development? How does it relate to these other established biomarkers? When does it first occur along this AD time course? And does it actually play a causal role, or is it just a co-occurring risk factor? Okay, so as neuroscientists, we often take a very brain-centric view of Alzheimer's disease and other conditions. And we forget that the brain is not an island, but it's indeed connected to a peripheral body, and the brain communicates with the rest of the body. This is particularly important in the context of AD because many systemic health factors are risk factors for Alzheimer's disease. For example, physical inactivity, a poor diet, an imbalanced microbiome, infections or pathogens or environmental toxins are all risk factors for Alzheimer's disease. And one mechanism by which the vascular system may influence the brain is through blood-to-brain transport. Now, a healthy individual will be somewhat protected from peripheral toxins, peripheral immune signaling molecules, et cetera, by something called the blood-brain barrier. So throughout the rest of our body, the vascular system can communicate very well with the other tissues. It can transport substances back and forth between the blood and the tissue, but the brain is uniquely privileged in that it has this barrier that protects the brain tissue from the circulation. And this is so critical because the brain is essential for survival. So the blood-brain barrier is it's a semi-permeable multicellular interface between the blood vessels and the surrounding parenchyma. It comprises endothelial cells that are joined together very tightly by proteins called tight junctions, and it's provided functional support by cells called pericytes, as well as astrocyte end feet. And together, this vascular system works with the neurons in something called the neurovascular unit. Now, there can often be this misconception that the blood-brain barrier is like a brick wall. But it is not, in fact, like this fortress here where nothing can enter and nothing can leave. A closer analogy would be something like a security guard at a private event or a club bouncer. So he might have a guest list that allows only select people into the club, and then he'll kick out people who are misbehaving, right? So the blood-brain barrier is very much like that security guard. It's a functional barrier that maintains brain homeostasis by regulating the bidirectional transport of substances between the blood and the brain's extravascular space. Okay, so we know that blood-brain barrier damage can impair delivery of nutrients, oxygen, glucose. It can disrupt the clearance of amyloid beta or other waste products. It can also permit the infiltration of bloodborne pathogens, toxins, etc. Now, some studies have suggested that there are some subtle structural or functional changes that occur to the blood-brain barrier with age. However, findings have really been inconclusive, and there's some consensus that these age-related changes might in fact be due to underlying pathophysiological changes. And this is supported by the fact that much more profound alterations to blood-brain barrier function occur with various neurological disorders, including Alzheimer's disease and related dementias. Okay, so we can measure blood-brain barrier permeability in vivo in living humans using two general approaches. The first is with biochemical markers from biofluids. So a common blood-brain barrier marker is can be taken from the cerebral spinal fluid using a lumbar puncture or a spinal tap. And this marker is called soluble platelet-derived growth factor receptor beta. It's a mouthful, but basically PDGFR beta is a marker of parasite injury. And as I mentioned previously, parasites provide important functional support to the blood-brain barrier. Another common biochemical marker we can use is something called the albumin quotient. And this is essentially the ratio of albumin in the CSF to the serum. Now, albumin is a protein that is typically only found in the blood. So if we see a higher level than expected in the cerebral spinal fluid, that would indicate that the blood-brain barrier is leaking out blood-derived proteins. Okay, so over the last several years, various studies have used these biochemical markers to probe blood-brain barrier function in Alzheimer's disease and other dementias. And generally, there have been reports of increased BBB permeability, but findings have been mixed. So here, back in 2017, Jenna Leeds and colleagues reported that the albumin quotient was elevated for individuals with Alzheimer's disease or other dementias relative to controls or individuals with MCI. Another study 2 years later also found that PDGFR beta was higher for AD versus controls. However, they did not find any difference when using the albumin quotient. Similarly, several studies have looked at differences by ApoE4. And for those of you who are not familiar with ApoE, ApoE4 is the single strongest genetic risk factor for sporadic Alzheimer's disease. It's not a deterministic genetic marker. So if you have an ApoE4 gene, it doesn't mean that you are definitely going to get Alzheimer's disease. However, it does increase one's risk in a dose-dependent manner. And so studies using biochemical markers have found mixed results. So for example, this study by Halliday et al. In 2013 found that older adults with at least one copy of the ApoE4 allele had a higher albumin quotient. However, that 2017 paper found no difference by ApoE4 across any of the groups they looked at. Okay, oops. So now these biochemical markers are quite useful. However, they do involve a lumbar puncture, which is not particularly comfortable. They're also indirect and they're regionally nonspecific, meaning that they can't tell us where in the brain this blood-brain barrier dysfunction is occurring. So that is where neuroimaging comes into play. So currently there are two broad classes of MRI techniques that we can use to measure blood-brain barrier permeability. The first is called dynamic contrast enhanced MRI. This is kind of considered the gold standard, and it's also the technique that my lab uses. So I'll be focusing most of the remainder of the talk on this method. But briefly, DCE MRI involves a series of T1 weighted MR images. There's a short period of imaging before the injection of a gadolinium-based contrast agent, and then a longer period of imaging after this injection. We can then convert the T1 signal to a concentration time course, and then using a region of interest in either a major artery or a vein, we can measure something called an arterial input function, and we use that to infer the vascular versus extravascular contributions to the concentration change. Then using some fancy pharmacokinetic modeling, we can compute something called K-trans. And this is the transfer coefficient of the contrast agent from the blood into the brain tissue. And this is our key measure of interest that estimates blood-brain barrier permeability. So it's basically measuring how much of the contrast is leaking from the blood into the brain. And here I'm showing an example K-trans map taken from one of the ADRC participants here in our study. And this was an 89-year-old male with a mild Alzheimer's disease, and I believe he was also an ApoE4 carrier. Now, this method does involve contrast, and contrast can be contraindicated for some individuals, particularly people who have a history of kidney disease cannot have a contrast MRI. So in these cases, non-contrast methods would be preferable. So over the last few years, there have been some developmental efforts to develop water exchange MRI techniques. And there are a few out there that different groups are using. So there's a class of arterial spin labeling methods. So arterial spin labeling uses magnetically labeled arterial blood. And multi-echo ASL and diffusion ASL leverage differences in either T2 or apparent diffusion coefficients between the vascular and the extravascular space. There's also a technique called double diffusion MRI, which relies on differences in water diffusion between the vascular and the extravascular spaces. Now, I haven't used these techniques myself, but I've kind of understood from colleagues that they have a slightly worse signal to noise ratio, and you often need very large sample sizes to get meaningful effects. So DCE MRI does seem to be preferable, but I am hopeful that some of these water exchange methods will undergo continued development in the future. Okay, so over the last decade or so, there have been a flurry of studies using DCE MRI to understand blood-brain barrier dysfunction in Alzheimer's disease. And I'm just going to highlight a few here. So back in 2015, Montagna et al. Published this very nice paper in Neuron, where they conducted DCE MRI on a small sample of young individuals, cognitively normal, older, and older adults with mild cognitive impairment. They found that within the hippocampus, K-trans, or blood-brain barrier permeability, was elevated for older participants and was even more elevated for those with MCI. They did focus on the hippocampus, but looked at a few other regions as well, and only found this difference in the hippocampus, which they interpreted to mean or to suggest that the hippocampus is the initial site of AD-related blood-brain barrier breakdown. Now, a year later, Van de Haar and colleagues published another beautiful paper in Radiology where they conducted DCE MRI on a group of cognitively normal older adults and those with MCI or AD. And they, in fact, found higher blood-brain barrier leakage across the gray matter and across the white matter. So in contrast to the prior study suggesting that this AD-related blood-brain barrier breakdown is really a global phenomenon affecting the whole brain. Now, more recently, just a few studies have looked at differences by ApoE4. And again, Axel Montaña's group published this really elegant paper in Nature where they conducted DCE MRI in a large sample of adults over the age of 45 along the spectrum from cognitively normal to early AD. And again, they found that K-trans in the hippocampus and in the parahippocampal gyrus, which is also in the medial temporal lobe adjacent to the hippocampus. K-trans in these regions was elevated for cognitively impaired individuals, but they also reported that it was elevated for ApoE4 carriers. They did not find the same difference in other brain regions they examined. And importantly, they found that these differences were independent of amyloid, tau, or brain atrophy. Now, the same year, another group also published in Nature on a group of cognitively normal individuals and those with MCI. They also observed increased hippocampal blood-brain barrier permeability for ApoE4 carriers. But in contrast to the Montaña paper, they found no difference between MCI and controls. They also found no association with cognitive function. So I hope that so far I have shown you that the findings in the literature are very mixed. And to really more directly assess the current state of our knowledge in this space, I conducted a mini literature review over the last 10 years, and here I'm trying to summarize it. So the top portion here refers to studies that used cerebrospinal fluid biochemical markers, and these two bottom sections show studies using either DCE MRI or water exchange MRI. And the positive findings are indicated in red, negative findings are indicated in a black none, and factors that were not examined in the study are marked with just a dash. And I hope you can appreciate that there's not really much consensus across most of these associations here. And just to briefly walk you through it, the strongest evidence is really for blood-brain barrier dysfunction in people with MCI or Alzheimer's disease. There's more limited evidence and inconclusive evidence for an association with ApoE4. There's relatively weak evidence for any association with age or AD pathology, including amyloid and tau. There's been very little examination of sex differences, and I didn't come across any studies looking at differences by race or ethnicity or regional specificity of AD-related BBB dysfunction. And only a few studies have looked at MRI measures of brain atrophy, and they've so far reported that AD-related blood-brain barrier breakdown is not accounted for by atrophy. So there are a lot of outstanding questions in the field, and some of the important ones, in my opinion, I'd just like to highlight here. So there's strong evidence that blood-brain barrier breakdown occurs at least by the MCI stage. However, associations with amyloid are pretty weak, and there's only limited evidence for an effect of ApoE4. So this raises the important question, when along this early AD cascade, MCI or earlier, when does blood-brain barrier dysfunction emerge? And this is a really important question in terms of whether BBB dysfunction could serve as an even earlier intervention target or preventative target for AD. In addition, most studies have either examined a priori regions of interest in the medial temporal lobe, specifically the hippocampus, or they've taken a global approach looking at the whole brain, gray matter or white matter. And so it's still not really clear whether early blood-brain barrier breakdown is localized to the medial temporal lobe or more globally distributed. And studies have suggested that findings of blood-brain barrier dysfunction aren't attributed to atrophy, but studies have not really addressed the question of whether blood-brain barrier dysfunction may actually contribute to neurodegeneration. So to address some of these outstanding questions, we're currently conducting an ongoing study in collaboration with the UCSD Shiley-Marcos ADRC. And today I'll be presenting preliminary data from 55 participants. This includes 44 who were diagnosed as cognitively normal, and 11 who were cognitively impaired, including 9 with MCI and 2 with mild AD dementia. Participants were intentionally enriched for AD pathology, and 70% were amyloid positive. They were also enriched for AD genetic risk. 55% were APOE4 carriers, and this is much higher than national averages of around 20 to 25%. As part of the ADRC longitudinal study, they underwent consensus diagnosis. They also underwent a multi-domain neuropsychological test battery. And the majority of participants, 50, had amyloid characterization. So 14 underwent amyloid PET. 32 had CSF measures of A beta 42 to 40 available. And for those without PET or CSF markers, there were 4. And we inferred amyloid positivity from plasma p-tau 181. And finally, they underwent APOE genotyping. Okay, so we administered an MRI protocol, which included a high-resolution structural T1 scan. We also administered a DCE MRI scan to measure blood-brain barrier permeability. And for those who are interested, the protocol included a 3-minute baseline, followed by a 15-minute post-contrast scan. I had a 20-second temporal resolution. We also administered a multi-shell diffusion MRI scan. And unfortunately, I don't have enough time to go into this in detail, but briefly, we used this diffusion MRI scan to compute brain microstructure measures from restriction spectrum imaging. So briefly, RSI, restriction spectrum imaging, is a multi-compartment diffusion MRI model that models diffusion orientation across a spectrum of length scales. This allows us to measure different diffusion patterns that correspond to different tissue properties. So specifically, we get measures of restricted diffusion that correspond to intracellular diffusion. This can be either within cell bodies or within axons and dendrites. We also can output hindered diffusion, which can either reflect the extracellular space or large cell bodies. And finally, free water, which corresponds to the cerebral spinal fluid. Our DCE images were processed through the Rocketship software package using the PATLAC model with a vascular input function in the superior sagittal sinus. And we defined our regions of interest using FreeSurfer and AtlasTrack. Okay, so our first question, as I mentioned, there hasn't been much examination of regional differences across the brain in blood-brain barrier permeability. So to start off, we just wanted to understand the regional topography of permeability across the brain. And so here I'm plotting K-trans for various brain regions. And if you focus just on these leftmost 2 bars here, we can see that gray matter permeability is higher than white matter. And this has actually previously been reported in other studies, which was a good sanity check for us. Then if you look at the remaining bars, I've color-coded them by brain lobe or brain region. And you can probably appreciate that there's not really any systematic clustering across the different brain regions, indicating that there aren't particular lobes of the brain that are more susceptible to higher permeability in general. Um, However, when we look more closely just at some of the brain regions here, we did note this interesting finding that the entorhinal cortex had the second highest value of BBB permeability. And I remind you, this is within an older population that is highly enriched for preclinical and prodromal AD. When we look at the hippocampus, which prior studies have really focused on, we don't see that overall the permeability levels are that high. So kind of integrating together what we've seen from the past literature and kind of these regional findings, we focused on 4 regions of interest for our analyses. So first, to test the hypothesis that AD-related breakdown is localized to the medial temporal lobe, we focused on the hippocampus, given prior research, and the entorhinal cortex. And I should mention, for those of you who are not familiar, the entorhinal cortex is one of the earliest sites of tau accumulation and a neurodegeneration in Alzheimer's disease, so we're particularly interested in it. And then to test the competing hypothesis that AD-related blood-brain barrier breakdown is more widely distributed, that it's a global phenomenon, we also measured our MRI measures in the cortical gray matter and the white matter. Okay, so what did we find?
Can I have a question about the last
Yes.
slide? It looks like, are these free surfer regions or what atlas did you use?
Yes, we use free-surfer regions, the cortical parcellation and then the subcortical ASEG.
Okay, and it looks like you've got most of the subcortical are on the left. That's what I'm
Yes.
seeing. Okay, so you've got the subcortical tends to have less of a problem with blood-brain barrier than the cortical. Would that be a way of thinking about it?
You know, I don't exactly know how to interpret this. It's maybe indicates less of a problem. I don't know how much we can interpret from just average permeability level. Physiologically, I don't fully understand, like what would make permeability higher in one region versus another beyond damage, right? So there could be functional differences. So it could be that, this is a healthy, normal level for that area of the brain. Or it could indicate that, caudate nucleus accumbens are really protected from blood-brain barrier dysfunction. So I hesitate to say, regions on the right are problematic, regions on the left are healthy, but that's one possible interpretation.
Gotcha. And then for the cortical region, so entorhinal there is just going to be the cortical part of it. It's not the white matter underneath it.
That's correct.
Okay. All right. Thank you.
Okay. So to start with, we found no associations between K-trans in any of our regions and age. Um, and to my great surprise and alarm at first, we also found no difference between our cognitively impaired and our cognitively normal groups. And at first, this was, when I first analyzed these data, I thought, oh, there's something wrong with our data. But when I thought more about it, um, I suspect that this is due to the fact that A, we have a small sample of cognitively impaired individuals, and B, which I think is probably the driving factor here, our cognitively normal group is very highly enriched for amyloid pathology and ApoE4. So they are not really a pure control group, but they're very highly represented by preclinical AD. So I think what we're really looking at here is there's not a significant difference between high risk or preclinical AD and cognitively impaired. Similarly, we also found no difference between amyloid carriers and amyloid positive individuals and amyloid negative individuals. Okay, so moving on, we did find that ApoE4 carriers had higher K-trans across the cortical gray matter. And this was significant after adjustment for age, sex, and amyloid. And I'd like to highlight some work that my fantastic postdoc, Serafina Soldiers, is working on right now. She's trying to better understand the regional distribution of these ApoE4 related differences. And here you can see the beta value for the ApoE4 difference plotted across the cortical gray matter, and these are the free surfer-derived regions. And there's this interesting pattern where it seems to be very superiorly located. So you can see the hot spots in the yellow and light green tend to be really superior frontal and parietal. And so we're still kind of working through hypotheses about what could be driving that. But stay tuned for an upcoming publication from Serafina on that. Now, when we focus just on the cognitively normal subgroup, we find that this ApoE4 difference is really driven by the amyloid negative individuals. And so when you look at these four groups here, you see that really the only group that is not showing elevated K-trans are the ApoE4 non-carriers who are amyloid negative. So the interim conclusion so far is that there seems to be ApoE4 dependent gray matter blood-brain barrier leakage that appears even in the absence of measurable amyloid or cognitive impairment. Okay, so next, we wanted to understand whether this blood-brain barrier permeability is related to neurodegeneration. So we looked at associations with our RSI measures of brain microstructure, and I have a little cheat sheet here if you forgot what the different RSI metrics mean. But across all participants, we found that higher entorhinal permeability was associated with increased free water, which is a marker of cerebrospinal fluid, and higher white matter permeability was associated with lower hindered diffusion, which is hindered is somewhat of an ambiguous measure, but most often it seems to correspond with the extracellular space. Now, when we look at interactions with amyloid, we find that there were significant interactions for associations of entorhinal cortex blood-brain barrier permeability and microstructure. Specifically, amyloid positive individuals showed correlations between permeability and lower neurite density, so axons or dendrites, and higher free water. Similarly, we found an interaction with ApoE4 such that ApoE4 carriers only showed a correlation between higher entorhinal permeability and higher free water. Now, unfortunately, we weren't adequately powered to look at three-way interactions between K-trans, amyloid, and ApoE4. However, when we do stratify by amyloid and ApoE4, we see this pattern of highest correlations for amyloid-positive ApoE4 carriers, and the weakest associations for amyloid-negative ApoE4 non-carriers. Okay, so we're doing okay on time. So to summarize so far, or to summarize, this is the end. I have no more data to show you. In our sample enriched for Alzheimer's disease genetic risk and pathology, we found that blood-brain barrier permeability was not related to age, cognitive impairment, or amyloid beta. However, ApoE4 carriers showed cortical gray matter blood-brain barrier breakdown, even in the absence of detectable amyloid pathology or cognitive decline. And notably, this was distributed across the global cortical gray matter, and we found no evidence that it was localized to the medial temporal lobe. And notably, and in my opinion, this is kind of the most exciting part of our study, we found novel evidence that blood-brain barrier breakdown is associated with abnormal microstructure in the entorhinal cortex and the white matter, and associations within the entorhinal cortex between blood-brain barrier breakdown and lower intracellular diffusion and higher free water were most pronounced for amyloid-positive ApoE4 carriers. Okay, so integrating all of our data our findings suggest that there's a widespread increase in blood-brain barrier permeability that may occur early along the Alzheimer's disease time course in an ApoE4 dependent manner. And this ApoE4 related blood-brain barrier dysfunction may promote neurodegeneration, particularly in the presence of amyloid pathology. Okay, so where do we go from here? So I'd like to acknowledge some limitations. So first of all, DCE MRI measures leakage of a contrast agent. This is a high molecular weight molecule, and therefore it's measuring only one aspect of blood-brain barrier permeability, and it may not be able to detect other functional changes or more subtle leakage. As I previously mentioned, our cognitively impaired group was rather small, and our control group was really highly enriched for AD pathology and genetic risk. So we were most likely limited in our power to detect differences by cognitive status. And we are continuing to recruit right now. And so I'm hoping to get kind of a larger, pure, cognitively unimpaired group to better probe that question. Some future directions, as our sample grows, we'd really like to examine differences by sex as well as race and ethnicity. And in addition to the genetic factors that we're looking at, it'll be really important to understand the role of modifiable risk factors, particularly vascular or metabolic risk factors. And given our really interesting finding of this association between blood-brain barrier breakdown and microstructure in the entorhinal cortex, which is a site of early tau accumulation, we are hoping to get funding for a follow-up study examining the roles of tau and inflammation in these associations, to better understand how they might be involved in mediating or modifying effects of BBB-related blood-brain barrier dysfunction on neurodegeneration. And then lastly, I'd just like to highlight my current R01 that is expanding on these findings to understand the effects of long COVID on blood-brain barrier breakdown in older adults. And we're particularly interested in the question of whether long COVID could interact with either pre-existing risk for AD, pre-existing AD pathology to accelerate the progression of AD, or if it could, yeah, basically how it can interact with AD genetic or pathological risk. And we are actively enrolling right now. So if you or anyone else you know is interested in participating, we are recruiting participants aged 50 or older, either who have long COVID with persistent cognitive complaints. We're also enrolling cognitively normal individuals who do not have a history of long COVID. So please reach out if you're interested, or feel free to pass this along to anyone else who might be interested. And then lastly, I'd just like to acknowledge everyone who contributed. So my current and prior lab members, all of my fantastic collaborators, funding that made the work possible. And of course, a major shout out to all the participants for your time and your effort in the MRI.